Thank you, HUD. All right, everyone hear me in the back? All right, I speak very fast, I'm sorry, but it's just because I'm very excited, and if you want me to repeat it slowly afterwards, just come find me. So I'm Ethan. I'm honored and pleasured to be here. I've lost count how many of these conferences I've attended, but I consider you all a kind of second family. And I want to also do a mostly data light talk and really engage in some storytelling. And the title of my talk was a little bit more verbose than this, but I simplified it. And I really want to talk about in decade-long spans, because that's how long we've been at it, and that's potentially how another decade at least in front of us. So you've already heard a little bit about drug repurposing. I'll talk some more about that. I would say that that has characterized the last decade or so, but I think we're on the cusp of a transition where we're going into a drug discovery mode. And I think the playbook for drug discovery, especially targeted drug discovery, to make the equivalent of small molecule gene therapies, we are on the cusp of a decade of breakthroughs, I think, in this area, and I think CDG should be at the forefront of benefiting from these type of targeted therapies. But we wouldn't be in this position to exploit targeted drug discovery if we hadn't built a really solid foundation in drug repurposing. So I want to recap a lot of different data, a lot of different stories. That the underlying data is available either in publication form, in peer-reviewed journals, or on Substack, and I can get into those citations later. But I really want to just convey a lot of the work that Perlara has done in collaboration with many of the folks here, in particular Dr. Ava, may I call you Dr. Ava? And many others here, clinicians, families, researchers. So hopefully you'll hear maybe a shout out, and if you don't, I apologize, I just couldn't cover everybody. And then I'll end with a live demo and some other teasers of what I think this era of drug discovery that lies before us. And I'll just say I don't have a proper disclosure slide. Apologies, but I guess I'm doing my disclosures by my X accounts. So Perlara is my main disclosure, and then I just launched a new effort called 1,000 Cures with my co-founder who happens to be here. If you want to talk about that, we can chat later. Okay, next slide. Oh, I can advance the slides. My goodness. So Perlara was started in 2014 as the world's first biotech public benefit corporation. Like many companies that survive this long into their 13th year, you have to undergo a few transformations. So the first version of the company I call Perlara 1.0. Maybe you recognize the special lady in the middle there. I really started Perlara for a very simple reason, because at the time there was really no companies that were really serving the rare disease space, and in particular weren't really serving rare disease parents and families and letting them really drive the process, which I think I've seen now time and time again how that has worked. So we initially started with this concept of a Pearl Quest, a collaboration with intrepid rare disease families who have a founder profile, who are entrepreneurial in spirit, who do not take no for an answer. And we launched a number of these collaborations with various family groups. Some of them are here today. I already mentioned Holly and others. And we ran that for a while, and it culminated in a couple of discoveries, and I'll talk about one of those in particular. But that model sort of ran itself out, and we transitioned to a different model, which was what I call Perlara 2.0, which was to double down on this concept of providing research as a service, in particular drug repurposing as a service for families, and where families could retain control of the IP and retain control over the direction of the project. We were really providing the laboratory rails and know-how to be able to accelerate them into the clinic. And that chapter has closed, and I'll end my talk with Perlara 3.0, which is now deprecated this consulting drug repurposing as a service model. I guess if you're still super interested and motivated and you want to talk to me, just like with Clement, we cannot knock these out one at a time anymore. We have to be more systematic, but we still will take on a few cases here and there. But for the most part, we are transitioning now to being a grant-funded company. So Perlara received an SBIR, Small Business Administration grant, for $2 million last year to really focus on drug development for inherited metabolic diseases, including CDGs and mitochondrial diseases. And if you really want to dig in some more, we have a publication venue called Substack. Maybe you've heard of this. You can find us at cureodysseys.com, and you can see case study after case study. And I'll mention some of the data and some of the folks, and I'll try to call them out, although I have this really bright light in my face, so I can't really see all of you. But if you really want to dig into some of these, you can tuck into our Substack, and there are dozens of posts where we go into the data and go into going from a drug repurposing screen to a hit to N of 1 data. And I would be remiss if I did not give a shout out and call out to my business partner, Holly Carmichael, and Maggie Carmichael, who was the N of 1 pioneer for the Apowerstat study. We created a company together called Maggie's Pearl to pursue commercial development for Apowerstat. As Ava told you, we failed, and for that, I apologize, and maybe that sounds weird, but I think I was as devastated as Ava was and as everyone else was, but I think the silver lining is that we learned so much from this that the next time anyone in this space, any drug developer, not just Perlara, any drug developer in this space will benefit from the sacrifices that all of the participants in the study made. So if you were a participant in that study, I thank you again. And it was not in vain. We have learned a lot. And this is unfortunately the brutal reality of drug development, but we're trying. There's no luxury for a pity party. You don't have that luxury, so we don't either. And we're gonna get back up on our legs, and we'll have a path forward, I think, soon. So this concept of a patient avatar, I think, has already indirectly been referenced through Clement's talk. But Perlara, from the very beginning, was very invested in this idea that you could create patient avatars and use them for drug screening. Drug repurposing is just one type of drug screening. And we realized that you could take advantage of these very simple model systems. You could install the mutation that is causing the gene in a affected child. You can install that mutation into a yeast or a worm or a fly if it has that corresponding version of the human gene, which for most metabolic disease genes, they are conserved in yeast and worms and fly. And then we do a very simple experiment. We sprinkle, as Ava said, we sprinkle a little bit of 8,000 different drugs onto these mutant avatars, and we just wait and watch and see who grows. And it's very simple. So the yeast don't grow, and all of a sudden they do. The worms don't grow, and all of a sudden they do. The flies don't develop, and all of a sudden they do. And that's the first tier of the process. And then there's other layers of validation. You've heard of some of them. We use skin cells or fibroblasts from the patient. We can even derive these organoids, these mini brains in a dish, and we can test the hits that emerge from these so-called primordial models, these patient avatars, and really assess which ones have the legs to go all the way into humans. So that's summarizing. There's a lot more details and a lot more you can go into this, but I'll just suffice it to say that Perlara has focused extensively in the last few years on yeast as our primary engine. I know Clement has focused mostly on fly, but we actually have converged on very similar types of findings. So it really doesn't, I think, matter which organism you're starting with. As long as you see convergence, then I think you're onto something. So I will talk about two types of convergences. One will be pharmacological and one will be biological. So I'm a biologist, but was trained in a chemistry lab, so I like chemical structures. Maybe some of you are getting very bad memories or from organic chemistry, but I'm not gonna give you a pop quiz here. I just want to show you a couple of the learnings at a very high level. So we performed a series of screens for PMM2 CDG when we engaged on a Pearl quest with Holly and her family way back in 2017, 2018. And the net result of these screening activities are summarized on this slide. So in the yeast screening campaign, we identified one natural product, which I'm showing you here. And then in a parallel worm screen, where the worms also had the PMM2 CDG, or a worm version of PMM2 CDG, we discovered a different molecule. But as you can see, and I've aided you with my illustration here by coloring in the part of the molecule that I want you to pay attention to, and the part of the molecule that this so-called pharmacophore that's conserved, you can see that right away, the yeast and the worms, they didn't give me the same answer, but they gave you a similar answer. And then we were scratching our heads for a bit in the beginning here, wondering what do we do next, because that first compound that came from the yeast screen is the active natural product in cinnamon. So either, I guess, take a boatload of cinnamon, which I think maybe you considered doing. That second compound from the worm screen is a natural product from some plant whose species name I forget, but also not something you could readily order from Amazon. And then the light bulb moment was figuring out that there was a drug, a synthetic drug, that happened to have similar pharmacological features or similar structural features, and that's what we ended up focusing on as a Apowerstat as the actual clinically actionable path forward, because we couldn't move forward with those supplements. And we obviously wanted to understand what did these three molecules actually have in common, and the answer was aldose reductase. And so why does this orange pharmacophore appear in the first place? And I think what we've come to the conclusion is that we're really seeing a mirror, a reflection of natural products, natural metabolites that are funneling and cycling through metabolism, And the molecules that we're pulling out are somehow echoes of these molecules, which suggests that they're engaging with the same protein binding partners that are engaging with those endogenous metabolites. They might also be engaging with these natural products, which have evolved to mimic these naturally occurring metabolites. And so I'm showing you here glucose in two of its forms. It's equilibrating and exchanging between this linear form and this cyclized form. And then when it's phosphorylated, it produces this species glucose-6-phosphate. And that's where you can really see this chemical mimicry appear. And so in the Perlstein case, you've expanded the pharmacophore, you've wedged in this heterocycle in between, you've sort of stretched out the pharmacophore. In the case of the perporogalin, you've actually sort of rigidified the molecule. In the 4-hydroxymic acid molecule, that tail with the OH on the left is kind of able to sort of dangle a little bit. But in the middle compound, it's sort of fixed in place, like when you have a cast on your arm and it's sort of locked into position. So these patterns where we'd see the same pharmacophore, sometimes in its stretched out form, sometimes in its cyclized form, and sometimes in a synthetic form that still has echoes of its origination, but is now a distinct molecule, the parsimony suggests that they're all sort of doing the same thing, and they're all interacting with proteins that are dealing with glucose-6-phosphate and glucose metabolism. So was this just a fluke with PMM2? Was this just something to do with yeast biology? The answer is definitively no. I'm just going to summarize two other examples of this pharmacological convergence. This is from SRD5A3 CDG. Shout out to Vijay if you're somewhere in the audience, whose foundation supported this work. And in this instance, this is a different naturally occurring metabolite called mevalonate, which also switches between this linear straight form and this cyclized form. And what I'm showing you below are the 6 compounds were the top hits that came out of an SRD5A3 worm screen. So these were compounds that rescued a worm that was given SRD5A3. And what you see is the same pharmacophore expressed in all of these molecules, and the same themes, where sometimes the molecule is expressed in its linear form, kind of attached as a handle to this other sort of building block. Sometimes the pharmacophore is cyclized. Sometimes it's like a musical note, it's doubled, like in the hypocrelin compound, the one on the lower right. You can see this doubling. So this really started to suggest to us that there were these common themes here, and that there were these central metabolites, these core, maybe keystone metabolites you might want to call them, that have inspired a lot of molecular mimicry. And the reason why you have the mimicry is because these molecules are all sort of interacting with the same proteins that are built to bind and to manipulate these metabolites. And yet here's another example of pharmacological convergence. This is for GMPPA. Shout out to Diana. Where are you? Right there. So this is actually, I can't claim credit for the screening results. These are results from Clement's lab. His fly screen, where he created a fly with GMPPA, yielded 3 hits, and really 3 hits only, and these are the ones. And maybe you can see here the commonality. It'll be a little bit easier to show you on a black background. But here you can see that we have molecules that converge on essentially a completely independent pharmacophore, this one involving more mimicry of a sterol, of a cholesterol molecule, than glucose or mevalonate. But I think you've got the picture now that the more we do these screens, the more we find that there are these keystone metabolites or sort of privileged pharmacophores that keep showing up again, because those are the molecules that are being manipulated by metabolism. And if you want to manipulate metabolism, you have to engage in some amount of mimicry. Okay. So I talked a lot about these pharmacological convergences. What about biological convergences? In other words, we've now looked at 35 different inherited metabolic diseases, including about half a dozen CDGs. About half of them are mitochondrial diseases, and we've modeled them in different ways. In some cases, we have genetic mutant models. In other cases, we have pharmacological models where we use a naturally occurring poison for, say, complex 1 or complex 2 of the electron transport chain. And we're just looking at a correlation graph here where we're asking, just like Clement showed in his slide a few talks ago, what are the shared rescuers? What are the shared molecules that rescue these phenotypes? And what you see, you start to see, and I'll show you in my demo in a second, but you're starting to see these neighborhoods and these clusters forming where different disease models are grouping together in spite of not necessarily being labeled the same. So we're seeing, connections between mitochondrial diseases and CDGs. And we're told, or at least the way we set things up is that, well, that's one community because your genes are these, and you're another community because your genes are these. But I think our results, and I think Clement's results suggest the same, that we should be thinking about inherited metabolic diseases the same way we think about autism spectrum disorders. We should be thinking about metabolic spectrum disorders and less fixated on the gene that is affected and more fixated on the pathway and the effects that the ripple effects have on metabolism. And that's where you start to see these non-obvious connections where, okay, if you have two genes in the same pathway, like, for example, all these MT genes or all these aminoacyl tRNA synthetase genes, they all sort of enzymes that do the same thing. And so you expect some kind of intrinsic similarity between them. But we also see these non-obvious connections where a CDG looks like a mito disease, or where this particular mito disease looks more like this other subgroup, but not this other one. So I think this all reinforces this idea that we should think about metabolic disease as a spectrum, the same way we think about autism spectrum disorders. Okay, so I have a surprise for you. I'm going to switch over to a live dashboard. So this is the debut of Perlara 3.0. And let's see if I've got control over this screen. Okay, so you can access this. This is all public now. You just go to perlara.com and you click on Pipeline up here. And it took about 3 or 4 years to generate all this data. And it took Claude 30 minutes to put together this dashboard. So I think it gives you a sense of how fast AI is changing. I mean, literally by the week. And where the real bottleneck in AI is the reinforcement loop. What are we telling the AI is the ground truth? You need experimental validation. You still need organic intelligence for that. So I'm not going to spend too much time here because you can all go off and frolic, especially if you want to nerd out. Here's your chance. But I'll just walk you through some of the features of this dashboard. And I think Clement has already teased you with similar type of data. And in our case, we were able, as I said, to look at 35 models at this point, and we've screened just under 8,400 compounds for each of those models. So let's look at this Gene Explorer view. We can now click here, and we can scroll down and look at what's your favorite gene. We just talked about ALG13. Why don't we pull up that? And so you can see the hits are listed here. The rescuers are on the left. The sensitizers, in other words, the molecules that make it worse, are on the right. You can scroll here and say, I want to see the top 10. I want to see the top 25, whatever your heart desires. And then you can start to look at some of these compounds in more detail, and that's in the Compound Explorer tab. So let's look at some of these over here, just for the heck of it. So we can click over here, and you see each one of the different disease gene models. There's 37 here, but there's a couple of replicates. And what we're looking at on the y-axis is a z-score, which is just simply a score of how statistically significant is the rescue or the sensitization. And in this case, we're looking at the top compound that came from the ALG13 screen, this guy right here. So we can go over here, enter its name, and then you can see, aha, wow, this hit was pretty selective. Really only rescued ALG13 and didn't really touch anybody else. Let's look at some of those other compounds. And as you can kind of see where I'm going with this, we see many, many types of patterns here. So here's a compound that was, as Clement said, there's a few sort of rock stars, or at least ostensibly rock stars, that seem to rescue across the board, but they also have some disease models where they're completely neutral. And so you can kind of go through this list, and you can see that there's many, many different types of compounds where you see, in fact, here's another one that's selective to ALG13, but then we also see cases where, let's do this one, where you see, again, rescue across multiple different disease models, and where it's not obvious that all of these diseases are actually really in the same pathway, so there's this biological convergence that defies the gene label. Okay, and then we just look at the sensitization side, so we can look here at a compound like, let's look at this one. And here, it's kind of a mixed picture. And in fact, there's a non-trivial number of the compounds that we looked at that have a mixed profile, where in some contexts, it's no, 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 don't do that. In other contexts, this might be a rescuer. And then in others, yet still, it's neutral. So interpreting these data, no single one human brain can assimilate all this information. So it lives here, but we're going to obviously have to keep using AI to extract all of the value from this. But I won't belabor the point here. You can look at all of the 8,000 compounds to your heart's content, and you can convince yourself that vast majority of compounds have no effect. And so when you do see an effect, it's interesting. And then we can do things like comparisons. So we can compare any of the two screens that we've done. So just for the sake of CDG camaraderie, let's do ALG11 and ALG13. We can rescale the graph here. So here's an example of a compound that's a shared rescuer, right? So if you're out here in this upper right quadrant, that means a compound is rescuing both of the different gene disease models here. And then you can start to play around and you can ask, okay, well, let's pull up, for example, this one, AFG 3L2, is a mitochondrial disease gene. And they're mostly uncorrelated, is kind of what you might expect. But then you start to see a surprise. Look at the upper left quadrant. All of a sudden, you have a bunch of molecules that make AFG, the AFG model, better, but they make the ALG 11 model, in this case, ALG 11 model, it's worse. I can go back to ALG 13. Same, and in fact, you see that same pattern here, where there is a group of compounds that makes one better, but the other worse, and so on and so forth. You can find essentially all quadrants of these plots end up being inhabited in some condition or another. And then I'll just sort of end here by saying, okay, we can look at all the genes, and we can say in blue are the rescuers, and in red are sensitizers. And it's just a count, okay? And it's a little bit crowded here visually, so let's just focus in on some of the individuals. So you can see there's, for each of these genes, and we can't predict why, sometimes there's an excess of rescuers relative to sensitizers. Sometimes it's the other way around. And sometimes you get very few rescuers, and sometimes you get many rescuers. So it's sort of, we have no real way to predict this yet, but you can sort of get a sense here that for every screen we've done, we've always found something. And then if you look at we have the compounds are de-identified here, but you can look here at pathway enrichments and see, well, what are the annotations of the mechanism of the molecules that are showing up as rescuers, and not surprisingly, they're enriched in metabolism. And then here's, I think, the most comparable plot to what Clement showed you, where we have 37 by 37. It's a little bit of compression here, so you can't see all of the legends. But again, it reinforces the point I made in the static slide a few moments ago, where you're seeing these blue blocks emerging. So these are shared rescuers, so compounds that rescue both. And so this is where we start to see this concept of biological convergence emerge, and we have to now drill in and ask, why does one particular disease gene model, correlate with another? And of course, there's just too much data here to summarize or for one brain to assimilate, but that's why we created this dashboard. Definitely looking to work with other folks, especially AI folks who want to help us extract meaning from this. Okay, two more notes here. As I said, we need to transition from drug repurposing to drug discovery. I think in many cases, for some communities, I know you might not even have tried drug repurposing, so definitely do that. But there are some among you who have already graduated from drug repurposing, and what's next? And as we saw from the power stat, it is a bit of a cautionary tale, because if you do not know who is going to respond and who doesn't, and you take all comers, you're likely to see the same outcome. And so we think the answer, or I think the answer to many of these rare genetic diseases are what I would consider small molecule gene therapies. So in the field of cystic fibrosis, there are a series of drugs culminating in a triple combo called Trikafta, which is essentially a gene therapy in a pill, right? Technically these molecules are called chaperones or correctors, but they are small molecules that bind to the mutant protein, and to Akhilesh's point, they activate that protein, or they restore its folding. We're not trying to inhibit these enzymes at all. We're trying to do the opposite. So one example of a group that I think has graduated from drug repurposing, and it was successful to some degree, and is now, I think, poised for drug discovery, is PIGN-CDG. So shout out to Susie Lamb, who's representing the PIGN Cure Collective. And here's another example of how amazing AI is. Working with Hiren, Susie's husband, and Hiren's childhood friend, who is at NVIDIA, we're able to whip together, and the URL, don't worry about this mumbo jumbo, the URL is alice.bio, ALICE.bio. And what you have here is essentially the starting point, I think, for a targeted drug discovery for CDG. And in here, we just populated with 5 different CDGs, including PIGN. But essentially, this model you're looking at here is interrogating many other data sources and many other sort of ground truths and other, other open source models that are, allowing for protein folding visualization and other things, all compiled in about 2 hours of compute time. This entire dashboard was whipped up. And so you can look at a particular mutation, let's say in PIGN, and it will run this process here. It will recommend molecules that the AI thinks could correct PIGN mutants. So some of this is slop. Obviously, you have to double check this. You cannot take all of this on blind faith. But I think we're at a moment in time here where, with a few hours of prompting, or a few prompts and a few hours of just waiting on compute time, you can essentially have a biotech company in a box. Now, this is focused on ultra-rare diseases, but I think you could imagine applying this to any one of the metabolic diseases, any monogenic disease, really. Because I think the future moving forward beyond drug repurposing is targeted drug discovery. It is gene therapy in a pill. And I think groups like PIGN are, I think, on the threshold to take this next step. But then eventually all of you will graduate to this point. And AI is only going to make the acceleration faster. So I'm very, very excited to see where things go. And thank you for your attention and would love to take questions.